Welcome everybody to this second part of this cycle that we are being organizing in jointly by the Institute of Complex Systems at the University of Barcelona and by the Catalan Association Complexitat.cat about the concept, not only the consequences, but about we can learn about COVID-19 and how we can explain some of these properties with using complex systems and what we can be done in the future relating this discipline applying to these pandemics. Okay, and just to remember the people that are attending this conference today that in about 3 weeks we have another one by Maria del Rio Chanona, that it's about economic consequences of the pandemics, and then later after the Christmas break or whatever we will have this year. We call it even Christmas, but some type of break that we will have. Then at the beginning of September, we will have the last talk of the second part of the cycle by Lucas Lacasa, also about, I don't remember exactly the title, but it's about reordering the people in the emergency rooms and how you can manage using modelization typically of complex systems. So then I give the word to Jaume Casademunt, who will introduce the speaker of today, Aleksandra Walczak. Please, Jaume.
Thanks, Albert. Well, it's a great pleasure to introduce Aleksandra Walczak today, and I want to thank her deeply for accepting this invitation to join this session, because I know she's very busy these days. So I'll say very briefly a few words about her. So Aleksandra studied physics in Warsaw University. Then she moved to University of California, San Diego for the PhD, specifically on the Center for Theoretical Biological Physics, where she studied genetic networks and glassy systems. She then became graduate fellow of the Kavli Institute of Theoretical Physics in Santa Barbara, where she studied population genetics and evolution. Then she was awarded a postdoc fellow position at the Princeton Center for Theoretical Science at Princeton University, where she studied again the gene regulatory networks and started to study the immune system. And since 2010, she's permanent researcher at CNRS in France, and she's specifically at École Normale Supérieure in the Laboratoire de Physique Théorique. Well, in recent years, she's worked on different aspects of the physical perspective on complexity in biology. As I said, in gene regulatory networks, in population genetics, but specifically in the study of the immune system. So this is essentially why she's here today. She has worked a lot on the collective behavior in the immune system, and she's contributed recently, in particular, to our understanding of COVID-19. So it's a pleasure to listen what she has to tell us today about it. So please, Aleksandra.
So thank you very much for the introduction and the invitation. Let me see. You see my screen now, full screen?
Yes.
Fine? Okay, good.
Yes.
All right. So as you know, you asked me to talk about COVID, so I will. I think it's going to be very different from the talks you usually have here. So there's going to be undoubtedly a lot of biology, so please don't hesitate to interrupt me and stop me. I'm going to try and be clear, but I know that sometimes, this may get too biological. So anyway, this work is done in collaboration with Thierry Mora, who's also at the and Asia Nervina and Misha Pogorelli, who are in Moscow, and the groups in Moscow and other collaborators, experimental collaborators who took part with it. But it's really, and with Mariam, who's also in Paris. So, but Asia and Misha really sort of spearheaded the experimental effort here. So let me explain to you what we did. So if you remember live last February, we could still go and do things like go skiing. So we got data from 2 people that went on a skiing trip and then came back home, didn't feel too well, quarantined, and right after the quarantine went in and gave blood samples. So we got blood samples from people who underwent a mild COVID infection last year, and we have longitudinal data from them, which basically means we have different time points. So what's the data? Basically, we take the blood, we sort out the T cells, and then we sequence the TCR repertoires. Separately, TCRs have 2 chains, alpha and betas. We sequence them separately, doesn't really matter. But basically what we get as a result of this is a list of the sampled, but let's say big list of TCR sequences that these people have. Okay, so what are TCRs and what are TCR sequences? TCR stands for T cell receptors. So you have an adaptive immune system. This you're probably aware of. Everybody's talking about antibodies. So the adaptive immune system has 2 parts. It's made out of B cells and T cells. Antibodies are soluble receptors of B cells. Okay, so we're going to be talking about T cells. So the other arm of the adaptive immune system, but it essentially works the same way. There's cells, the T cells, and they have receptors on the surface, and these receptors bind to pathogens. They basically bind to various molecules, but if it's a pathogen, they trigger an immune response. And the thing is that one cell has one type of receptor. It has many receptors, but one type. So to protect us against many different pathogens, we need a diverse repertoire of different T cells. And that's what the immune repertoire is. So if somebody's talking about the T cell repertoire, they mean the set of different T cells like that. And so how do we get the information? We get it now, there's different ways, but one of the ways is through sequencing. So as I said, you get a blood test, they pull out the T cells from your blood, they sequence them, and this gives you sequences for the different kind of T cell receptors you have. Now, so you understand this diversity because, well, first of all, I'm telling you, I can also tell you, you have of the order of 10 to the 9 different T cells. So that's huge. You have 10 to the, but you have 10 to the 11, 10 to the 12 T cells altogether. So that means that certain T cells come up in different abundances. And why is that? Well, if we have time, I'll get to tell you how they're generated. But for now, let's just assume that there's a way of generating these different cells. But after they're generated, if a T cell encounters an infection, so it recognizes a pathogen, it proliferates. That means it makes many different copies of itself, and the reason for that is that then it's easier for this T cell clone to recognize the pathogen. If it doesn't recognize anything, it dies. So there's some sort of dynamics going on here. But that's why when we talk about diversity, we need to talk about not just about the identity of the T cell, but also about the abundances. Sorry, is somebody trying to communicate with me? No.
Yes, sorry. Sorry. Some of us having problems with the screen.
Okay.
Now, perfect.
I'm having problems, or somebody's having problems?
No, I mean, when you refresh, sometimes it gets stuck and it doesn't refresh. Now it's okay. So.
Oh.
I don't know.
Okay. Okay. I just thought somebody was like chatting, and I don't see the chat. So. Okay, so we have a repertoire, we have all these different cells, we go in and, and since it's a dynamical system because a response is proliferation, so some cells go up, some go down. Then after stimulation, such as a response to COVID, which is what we'll be talking about, we can then track these changes of how the different cell abundances go up and down. This is what the data look like. Okay, this is like one T cell one T cell with one receptor, how it changes over time, and you see it's a mess. So you look at it and you don't really see much. So the first thing we did is we did something just to ask ourselves, is there any signal in the data? We took the most abundant clones and we did time-dependent PCA on it. Okay, and if we do that, we identify, the PCA identifies 3 main clusters. The purple cluster, which is the cluster of cells that do nothing, that don't respond, so we can forget about them. And then there's 2 types of response. One which we're going to call contraction, but in fact it's contraction after an early response. So the response happened at or before day 15. That's the green line, and that's what we'd expect, right? These people had an infection. Clearly the immune system went to work 15 after the infection, and then we expect it to be going down. Surprisingly, we also see this later response, which is an expansion and a peak around day 37. So we were not expecting that, but we see these 2 modes in both of these people that I'm going to be calling Terry and Pat, just to give them names. Obviously, those are not the real names. But so there is some trend in the data, but the data is also very noisy. So you know, we wanted to do a better job, and we knew from previous work that we should be doing a better job. So what we did is we calibrated a noise model for this data. So in order to do that, we took data from the same person at the same day, and when we scatter it against each other, so we ask, you know, for each T cell clone, for each T cell, we ask how many counts do you see in one sample versus the other. So this should be a perfect diagonal, and you see there's a lot of dispersion, there's a lot of noise. So we basically need to say that whatever data we see, this is going to be our diagonal. So what we do is we build a noise model that of the same day replicate data from the same person, so the thing that should be identical, that accounts for both biological noise and sampling noise. So we say, because we know from other data that maybe I should have shown you, because I know a lot of you like these things. So we know the clone size distribution is a power law. So if I do a rank, a probability versus rank plot of any of this data, I'll find the power law. So there's some clones that have high frequency and frequency. So that's the underlying data, but that's not what we're saying in our. So that's the underlying truth, but that's not what we're saying in the data. We're saying reads that come out of the sequencing machine, and before each clone, which is there with a frequency f, is turned into reads. Well, first of all, the cell actually has to be sampled, so that gives us Poisson noise from sampling, but also there's noise coming from differential expression. So as we know, where this experiment, I should say, is actually done on mRNA sequencing, not on DNA sequencing, and even if the receptor, I mean, even a cell with the same receptor can be expressing different numbers of mRNA, and we know these distributions are often long-tailed. So to account for that, we put in a negative binomial. So I'm mentioning this because I know some of you also like to think about things like mRNA expression and noise and that. So we put that into that, and from there we get from the original underlying frequency to the reads we actually sample through basically you know, but writing down the likelihood of seeing a given clone in the data, starting from frequency with these 2 sources of noise. And once we have that, once we have a noise model which we learn from data, then we can use it as a base for probabilistic inference of the response. So what does that mean? Well, we said that what a response is an expansion from frequency f1 to from frequency f2 to a frequency f1 times some exponential factor, because these cells are proliferating. Of course, we could consider different models too, but this seemed the simplest. And we said that we have a prior saying that, well, even if a cell is stimulating, there's some fraction, or in any kind of cells we're looking at, we have some fraction of non-expanding cells and expanding cells at a given time point. And then we calculate the posteriori probability, and we call an expanded clone if it has a large a posteriori probability of being expanded. So if we apply this to our data, we get back the two trends for the two people. We get this, what I previously called contracting. So note the change in the x-axis, which is time. Before I only showed you points from 15 on, from day 15 on, and I showed you this green line that went down. So this is the same thing. So that from day 15 to day 85 post-infection, there's a set of clones that go down. And then there's a set of clones that initially go up from day 15 to day 37 and then down. So that was the yellow trend. So we recovered the green and the yellow trend. You may be asking yourself, what are these two? Why do I now have these 2 points? So the interesting thing about this data is that we actually have data from these 2 people before the skiing trip. We have pre-infection data, so we know the repertoires at year -1 and year -2. So this is quite unique because it's as if we got data from them and then sent them on a skiing trip and hope they get COVID, which of course is not what happened, but we shall use this. But that's how we also know that this is a real expansion that happened around here, because here these cells are at smaller frequencies. So we see a large expansion. We also can then, I won't show you tables of what are the clones that are actually responding, but we can run them through databases, and most of them are not known to be specific to known viruses in the database. There'll be some exceptions that I'll talk about. There's really actually there are 3 exceptions. I won't talk about the ones that are exactly matching, but out of the hundreds of clones that we're able to identify this way, there's a very small number that's known to respond to something. Okay, so if we look at the list of the ones that are responding, here I'm showing you just the data for this green peak, so the ones that have a peak at day 15, but it's the same for the other one. We see that a lot of them have what's called a memory phenotype. Okay, so why is this interesting? So immunologists know that there are some cells that acquire a certain flag, which basically means something like, I've seen an infection. And then the second time they see a similar infection, they can respond more quickly. So at the later days, we see that many of the cells go into memory. And this is interesting because this means that this basically provides immunity for the future. Now, there's different kinds of memory cells, and some of them give you long-term immunity. And we have, I don't actually know if I'll show you, but some of them do have this phenotype. So this property that they're going to give you well, we don't know exactly how long-term immunity is, but this is what's called long-term immunity. So you probably know in the press, people there were, I think now we're past this point, but there was this point where people were freaking out that the antibody immunity is not long-lived and goes away after a few months, and that's kind of to be expected. It's not terribly surprising, but here we're seeing that you can have immunity also through T cells, so the other soldiers of the immune system. But yet again, the maybe more interesting thing comes from the pre-infection time points. And we see that clones that we later identify as responding are already there a year and 2 years before the infection. So these people have pre-existing immunity. Maybe that's why they get a mild infection. Okay, here for the aficionados, the stem cell-like memory, the pink ones here, are the ones that have long-term memory. and this is really for the experts, what we see is a shift from central memory pre-infection. So only for PAD do we have a breakup into memory and non-memory pre-infection to what's called effector memory, so a more sort of active kind of memory. So it seems that cells that were there already in this, giving them immunity, then get activated and expand. Okay, but bottom line is they seem to have they don't seem to have, but they have pre-existing memory immunity. So the thing is that you say, okay, so you do the to the noise elimination, and you tell me that these are clonotypes that are specific to COVID. How do you know they're specific to COVID? Maybe these people got another infection at the same time, maybe something else happened to them, and that's a fair point. So what we did is, well, we got incredibly lucky, to be honest, but we used data from what's called a tetramer study, which is essentially a fishing expedition. So you take your COVID molecule and you put it on the end of like a fishing rod of a molecule that lights up if anything binds to it, and then you stick it into a pool of T cells, and basically some test tube with T cells, and you see which ones bind, and you pull them up. Okay, so the thing is, you have one COVID protein which you're putting on at the end of your fishing rod, and you're asking what binds to this one COVID protein. Now, luckily, Pat participated in such experiment at day 20 after infection, and this is Pat specific data. And so at day 20, they identified a bunch of T cells that respond to this specific COVID protein. And then we took those T cells and asked how the frequency changes in time in our data set. And and that's how we get the blue line. Okay, the blue line is the mean, and the points are actually the ones that are the fraction of cells that are identified. And what this shows you, the blue line, is again we get back previously the green line, the line with the peak at 15. So the peak of this response to this protein is at day 15, and what the dots in green are here are the ones that we independently computational study. And you see that we're basically getting nearly all of the high frequency clones, so all of the high frequency cells we're identifying. And this is a way of doing it in a patient specific way. So this way out of just longitudinal data, we don't need any external information. So the person can be very weird in many aspects, and we can still identify their response. And of course, so this is validation for one COVID protein. We're identifying it's a response to many other proteins, but it's validation that the response works. Okay, so sort of wanting to further understand and also validate that data, we turned to motif analysis. So first of all, we can put links between T cells that have similar sequences. And if we do that, and this is again plotted for our contracting data, we see that we have many more similar T cell sequences in our data than we expect in a random repertoire. And these are different kinds of immunological subsets. But in all cases, the data is actually above the random expectation. So our responding cells are more similar to each other than we'd expect, and we can, based on that, we can cluster them. So we cluster them, we put a link. If the T cell is 2 amino acids away from another T cell, we put a link between them, and we get clusters. See that there are different clusters for W and M, which is Pat and Terry, and that's not surprising because Pat and Terry have dissimilar HLA types. So again, for the experts, basically this means they have dissimilar machinery that presents COVID proteins to the immune system or any other proteins. So we don't expect overlap, and we don't in the motifs, and we don't see much. So again, we're able to identify this protein that we just looked at a second ago with the tetramers with our fishing experiment, but we also see other clusters. So let's have a look at those clusters. And the way we're going to have a look with them is turning to another study. This is publicly available data from a company known as Adaptive. So what they did is they did a large-scale fishing experiment. So this is called what they call a mirror assay, and they put many COVID proteins here in rows, and in columns they put different samples of blood, and they basically have like the signal when there's a response to this samples of blood with different TCRs, so with different T cells. So if a T cell recognizes a COVID protein, then they get signal. It's a little bit more complicated. There's some interesting combinatorial analysis of that, but that's the general idea. So at the end of the day, they have that are known to be specific to COVID proteins, and what's more, they know what COVID protein it's specific to. And so we take their data, and we take our data that we identified as responding in our analysis, and again build similarity networks. So we put links if the amino acids are too away from each other, and this way we're able to identify clusters, and we're able to link 32 of our clusters to specific COVID proteins. So now we know what these T cells are recognizing. So this is one example of a cluster identified this way, and this is the COVID protein amino acid sequence that it recognized. And in fact, this bunch of T cells correspond, and this is the logo of that T cells, correspond to 21% of Pat's immune response. So 21% of all his T cells are responding to this one specific COVID protein. Okay. What's more interesting is that these T cells that belong to this cluster were already there a year before the infection. And if we make one mutation in this protein, in this COVID protein, we get a common cold protein. So we had the common cold a year ago, and maybe that's why now this is such a strong response, because it's similar to what he's already seen. I mean, this is at the level of speculation, but the fact that there is this overlap and this is the strong response, and it's a response to this protein, those are facts. Okay, so that was for the experts, that was all on CD8 cells. We can also, so there's two types of T cells, CD4 and CD8s. One of them are killer, the CD8s, the CD4 are what are known as helper cells. Doesn't really matter, you can just think there's two families. So we validated the CD8, we now want to so unfortunately, there's no mirror-like analysis for CD4, but there is a different study from the same group, from the same company, Adaptive, that basically took a bunch of people that had various COVID infections of various severity, and they just sequenced the repertoire. So basically the same thing we did, the T cell repertoires. And the same group a few years back did a study where they sequenced of the order of 1,000 people, just healthy people. So we have a sort of a healthy control. And so if we ask, so this time we're going to work with Terry. And if we look at Terry's motifs, we can concentrate on Terry's largest motif and ask, how is it overrepresented in the people who had COVID? So from this study compared to the control study. And it seems it is overrepresented, but as you can see, the data is quite noisy. Nevertheless, we were optimistic and we asked, can we identify people out of all these people that they have in the study? If we mix the healthy and healthy, can we identify who has and who didn't, basically by asking who has the TCRs, that T cells that correspond to Terry's motif. Let's just ask for one motif. And so we're looking at the true positive people who really had COVID versus people who didn't. And unfortunately, the answer is no. We don't seem to be able to do it. However, we then reminded ourselves about this HLA business. So remember, HLA is basically when a T cell, so a T cell has a receptor and you're trying to recognize a protein. That's what I told you. I kind of cheated because the protein gets presented on a scaffold, and the scaffold, so like a little bit of protein stuck on the scaffold, and the with the whole thing, the scaffold and the protein. So it's also important that the T cell has a good match to the scaffold. The scaffold is called HLA. HLA is also the thing that you need to get right if you're being an organ donor. So you want the person who's going to get your organ to have the same HLA as you, if that rings a bell for what it is. Anyway, so we said, but what if we use this information about the HLA for our purpose of diagnostics, figuring out who had COVID? Unfortunately, there's no HLA type in the COVID cohort. We don't know what HLA type they had. However, there's a very smart computational way of inferring the HLA type of a person. You basically take some data where you know the HLA type of the person, and then you look at the overlap of these people's T cells with the overlap of T cells, any T cells now not responding in the person you're interested in, don't know the HLA type. And thanks to that, you can infer it computationally. This is a method from DeWitt and Phil Bradley. So we used that. We did it. First of all, we were correctly able to identify Terry's HLA type, so that's good. And then when we asked if we now only people have this HLA type as Terry, can we figure out whether they had COVID or not? Now our ROC curve looks much better, and the answer is essentially yes. So we have, thanks to this motif analysis, we have a diagnostic tool. Now, I think I've been talking for 30 minutes, correct? Can you hear me?
Yes, you're about 30 minutes.
Hello?
Yeah.
Less than that, yes.
Okay.
You
So
have
maybe 10 more minutes?
Yeah.
So I'll okay. So okay. So I'll try. So I'll just show you one more result, but I need to do a little bit of build up for that. So okay. So we already went through this, that it's diverse, 10 to the 9 unique receptors. So where does it come from? Diversity come from? So this diversity of a receptor is encoded in this very funky process, which is called VDJ recombination. Each receptor is made out of 3 parts. But generally that's the idea. That's called, they have names. They're called V, D, and J, these 3 parts. And to get to the order of 10 to the 9 unique receptors, what happens is that you don't actually have DNA encoding for each receptor because you don't have enough space for this DNA. So your DNA gets edited in the cells. You have some number of templates for V, templates for D, and templates for J. And then you pick one V, one D, and one J, and you cut the other DNA out, okay? And so basically by putting together these building blocks, you're able to get to 10 to the 9 different combinations. Now, once you put them together with the numbers we have here, you get to the order of 1,000. So there's some insertions and deletions that come, nucleotide actually, deletions and insertions between the VD and the D and the J. And you have 2 chromosomes. So if it doesn't work on the first chromosome, you try on the other one. If it doesn't work, tough, but if it works on the first, that's great. You don't do the second one. But if it works on the second, but not on the doesn't work on the first, but works on the second, you have in your organism edited strands of DNA, 2 chromosomes, one which will be functional and one which isn't, which gets silent. Okay, and this is how you but at the end of the day, you generate, once you've generated a functional one that will work, be in frame, be a protein, you express it, and that's your naive repertoire. Okay, so this is what we sequence. And it turns out if you sequence, especially DNA, but it's also true for DNA, for mRNA, you see the in-frame reads. So in-frame just means it'll be a protein. So those were the ones that were probably functional, selected, and expressed on the surface, so real receptors. But you also see out-of-frame reads. So an out-of-frame read is a DNA sequence that will either have a stop codon or some other, well, basically likely a stop codon, so it will never become a protein. And so why is this interesting? This is interesting because these receptors were just they were not selected upon because they could not have been proteins. So this gives us insight into the generation process, and we used this. So what we did is, so this is our data, this is the process we're trying to describe, and what we're trying to say, what is the probability of generating any one of these sequences. So in order to do that, we built a probabilistic method, inference method. We take the sequences. For each sequence, we build a library of possible scenarios, and then we have a model. We calculate then, we assign weights to each of these scenarios, and then we calculate observables, such as the mean usage of a given V gene, mean number of insertions at a given junction, mean number of deletions. And then we have a model to recalculate the weights. And we do this self-consistently, and it converges. And so, I mean, all I really want to say is we have a way for figuring out what is the probability of generating any receptor. And I can also tell you that it's a very reproducible process. So it works the same in you and me and essentially anybody else in the world. So this is just to show you the insertion distribution over, so this is the probability of insertions. How many insertions are there at one junction, the VD in blue and the DJ in red? And you see it's very reproducible. The dashed line is the wrong model. I won't go over it. And it doesn't really matter. Basically, we have a model for now saying what's the probability of generating any receptor. And we can start asking questions with that, is how likely are you and I to share the same T-cell receptor? So since I know what's the probability with which they were generated, I also know the probability which I can also build a model for the probability with which they were selected if I want, but I can compare the probability, my random expectation, to what I actually see in data. And you see that it's pretty good. I mean, I can essentially predict how many receptors are going to be shared between two people. Of course, it depends on, you know, because it depends on how deep you sequence, how many sequences you have. But all I'm saying is that if I know what's the probability that you and I will share a receptor. And so with T cells, and then we can ask, so okay, so if I know the probability between with which you and I will share a receptor, I also can calculate how many receptors you and I are likely to share. And that's what this is. So the data is in blue, the prediction in red, and you see that we can predict the number, you know, the number of shared sequences between, well, 10 people, 100 people, and this is this very large data set with the order of 1,000 people. But we can still predict how many T-cell receptors they will share. These are healthy people. Same for B cells. So remember, B cells are the other arm that I didn't talk about that produce antibodies. We can still predict how many B cell receptors people will share. However, get B cell data from people who had COVID. Well, this is the data and this is the prediction. So basically, people who had COVID share more B cell receptors than we expect by chance. So that suggests that they actually underwent, you know, they have a similar response. And from other studies, we know that B cells that respond. So okay, so there's a thing about B cells that is not true for T cells. B cells, when they respond, they also make many copies of themselves, just like T cells, but they also mutate. So they start, the copies start being a little bit different. And so you can imagine that, you know, it takes time to mutate. So the longer you wait, the more mutations you can see. It turns out that when people look at B cells that respond to COVID, they're very not mutated. They're very similar. So there seems to be a type of B cell that is shared between people that is pretty good at responding to COVID. But basically, there is something more than we expect by chance. Okay, I'll stop with that. This is other stuff. So I just try to show you how from longitudinal data, just by mutational methods alone, we can actually identify T cells, we can characterize them, we can validate them by other methods. And the sort of biologically interesting things we find from them is that there is pre-existing memory in at least these people. They did happen to have a mild infection, so now we're interested whether, you know, how general this is. Of course, this is a much harder study to look at, and we do seem to see stable post-infection memory. Okay, thank you.
I'm afraid I had some problems, and I think that some other people had also in refreshing the screen, but more or less I fixed it at some point. So maybe some other people have also missed some of the slides. I did. I could hear all the talk, but not really all the slides. Anyway, it's now time for discussion. If there are any questions or comments or.
So thank you very much. The talk was very nice, and I learned a lot of things. And I was curious about, well, one thing specifically. So you said you identify this motif, this CD4 motif, and then when you look at people who have the same HLA I don't know, receptor, I don't know what's the name for it. Well, anyway, they have the same type of HLA, right? Then the CD4 motif, just by looking if they have the CD4 motif, you can tell whether they have had COVID or not, right? So do you expect that in other people who have a different HLA, it's exactly the same motif, or is it going to be?
No, it's not the same motif. So this was for, you know, I showed you Terry's data, and I also said that Pat and Terry have very different HLA types. So we can do the same analysis for Pat, who has a very different HLA motif. Then we find what their what their motif is, and then we look at, it's done motif by motif. So then we look whether this motif is there in different people. And again, if we separate out only the people who have the same HLA as Pat, then we get a strong signal. Whereas if we look at everybody, the signal is not so strong. So that, and then I can take another motif from Pat or another motif from Terry and redo the analysis. So I can do it motif by motif. The information we're getting from this is that the motif alone is not enough to identify a person, but the combination of motif and knowing the HLA type actually is.
Yes, of course, because otherwise your curve would look much better even without considering the HLA. But I mean, are there? So the thing is, I was wondering whether you can use this as like a test, like a serology test, to know if people have suffered COVID or not. So I was wondering how many types are there going to be? I mean, are there going to be as many as H? I don't know.
No, that's a good question. So, okay, I mean that's a very good. So first of all, you know, you can use it, but it's still a very expensive test compared to serological tests, because, you know, you have to do repertoire sequencing, which is more expensive than looking at serum. So basically, you know, it's not going to be in your local pharmacy tomorrow, but as a proof of principle, it is possible. Now, the other point that you're completely right on is that the HLA locus is the most polymorphic locus in the human body. So there are many, so we all have 6 HLA types. So like when I give you this type, it's one of the types this person has. But so there are some that are very common. So for again, Pat has a very common HLA type, and Terry has a the one we looked at is actually quite rare. So by showing it works for Terry with these, you know, 1,000 people from a different part of the globe, we're showing actually that it can even work for very rare ones. But it's true, you'd have to, you know, you'd have to design it for, but you know, you don't need to get all, so you don't need to get the exact HLA match like you do for a donor transplant, meaning you don't need all the HLA types to match. You just need the, you know, you just need one.
Okay. Thank you so much, Aleksandra.
Thanks for the questions. No,
you're welcome. It has been a really interesting, fascinating, really. So thanks.